The cell cycle is the life cycle of the cell, during which the cell grows, prepares to divide, and divides, forming two new daughter cells. The growth phase is called interphase, and the division phase is called mitosis or meiosis and cytokinesis. This is our key. The pink X's are the paternal chromosomes, which go in descending order of size. The orange X's are the maternal chromosomes, which also go in descending order of size. The pink string is the spindle fiber, the yellow connect pieces are the centrosomes, and the yellow circle is the nuclear envelope. When the nuclear envelope breaks down, it will appear as a dashed yellow line. The green circle, not depicted here, is the cell membrane. The cell cycle during interphase, which includes the G1, S, and G2 phases, takes up 90% of the cell's life. During interphase, the cell goes through checkpoints at the G1 and G2 phases in order to make sure the cell is ready to continue on to the next phase of the cell cycle. The cell cycle also includes the M phase, which consists of mitosis and cytokinesis. The M phase of the cell cycle includes 4 distinctive stages of mitosis: prophase, metaphase, anaphase, and telophase. This is followed by cytokinesis. During the G1 phase, the cell undergoes growth. The DNA is stored in coiled strands of chromatin within the nucleus, and the centrosomes are stored in the cytoplasm. Near the end of G1, the G1 checkpoint checks to make sure the cell is ready to move on to the S or G0 phase. After the G1 checkpoint, the cell can either go to G0, where it differentiates and performs its normal functions without dividing, or it can go to the S phase, where DNA is replicated. During G0 phase, the cell stops dividing until it is signaled to begin dividing again by signals such as tissue damage or growth hormones. During the S phase, the DNA is replicated, creating 2 sets of identical DNA, which will later condense into sister chromatids. After the S phase, the cell moves on to the G2 phase. During the G2 phase, the cell continues to grow. In our model, we forgot to include the centrosomes. The cell also must pass the G2 checkpoint. The G2 checkpoint checks to make sure the DNA is replicated correctly and that there is enough cell growth. Once the G2 checkpoint is passed, the cell is ready to move on to the M phase, where mitosis and cytokinesis occurs. Mitosis is split into 4 sections, with the first being prophase. During prophase, the DNA condenses into visible chromosomes and the centrosomes migrate to the opposite poles of the cell. The centrosomes have thin fibers of tubulin near them. The DNA also shortens and condenses into identical pairs of homologous chromosomes, also called pairs of sister chromatids. These sister chromatids are held together at the center by the centromere. The nuclear envelope also breaks down during this phase. The second phase of mitosis is metaphase. During metaphase, the sister chromatids align along the center of the cell along the metaphase plate. They move via motor proteins along the spindle fibers to the metaphase plate. The mitotic spindle is also fully formed during this phase, and the centrosomes have migrated to the opposite poles of the cell. The nucleus is also no longer visible. During the third stage of mitosis, anaphase, the sister chromatids move away from each other at the centromere and move toward. During the third stage of mitosis, anaphase, the sister chromatids move away from each other at the centromere and move towards the centrosome along the spindle fibers. The chromatids are now called chromosomes. The tubulin, which is no longer useful as part of the track, breaks off, adds to the non-kinetochore microtubules, and extends the cell, giving it a distinctive oval look to the cell. In our model, we forgot to elongate the cell, and our chromosomes are facing in the wrong direction. During the last stage of mitosis, telophase, the spindle fibers completely disassemble and disappear. The nuclear envelope and the nucleolus also completely reform, along with the chromosomes uncoiling into chromatin. During this phase, cytokinesis has also begun, which creates a cleavage furrow in animal cells. In our model, we forgot to uncoil the chromosomes into chromatin and make the nuclear envelope completely formed. After mitosis, cytokinesis occurs, where the cytoplasm splits in. After mitosis, cytokinesis occurs, where the cytoplasm splits and forms two new daughter cells. In animal cells, a ring of microtubules forms around the center of the cell along the imaginary metaphase plate and pinches together until the cytoplasm splits into two. In plants, the cell plate continues to grow using cellular materials until it divides the cell into two new cells. This cell plate forms the cell wall. The cell cycle is regulated by various factors. Density-dependent inhibition and anchorage dependence limits the rate of cell division in normal cells by requiring there to be enough space for the cell to divide, and for the cell to be connected to a stratum, such as the extracellular matrix. The checkpoints also make sure that the cell is ready to divide, and that the conditions are sufficient for the cell to continue to the next phase of the cell cycle. MPF, also called maturation promoting factor, and cyclin fluctuate in concentration during the cell cycle. When they peak, the cell is ready to divide. CDK, also called cyclin-dependent kinase, remains at a constant concentration during the cell cycle. When the cell cycle loses control of cell production, cancer can emerge. Cancer is triggered by mutations in certain genes that control both the number of cells that are made and the genes that trigger apoptosis. One of these genes is P53. P53 is a tumor suppressor gene that checks cells at interphase checkpoints to see if there are any genes in the dividing cell that are damaged or mutated. However, when the P53 gene itself is mutated, a cell with damage to its DNA can pass the checkpoints and divide, leading to cancer.